Okay, let me start with a quick introduction of our speaker. Lennart is a PhD candidate at EPFL in Bruno Carreras' lab, where he works at the intersection of protein design and machine learning. Before he completed his biochemistry studies with a bachelor's and master's at the University of Heidelberg. And he is co-first author of the paper One-Shot Design of Functional Protein Binders with BindCraft, which is the paper that introduced BindCraft to the scientific community. And today, this is the tool that is going to present us. Lennart, thank you so much for joining us. The floor is yours.
Yeah, thank you so much. It's a pleasure to be presenting today. So we are going to be doing a little bit of like a mix of a workshop and a talk, given that we have like roughly an hour. So I will be going. So I hope this is, yes. So we will talk a little bit about like where BindCraft came from and what is the motivation behind. I will give like an example on what we have been doing with it. And then taking one of the examples that we have been working on, I will guide you through like a trial run, and we will discuss the different kind of settings and files that you would need to prepare if you would want to run BindCraft. And then we will go through a few considerations and observations that we have been essentially making post-hoc when we ran all these design campaigns. And then I will also, because I'm actually mostly a wet lab scientist by training, give a few notes on the wet lab for the people that come from the other side and maybe want to bring the first few designs into like a real experiment. So just to motivate us why we want to actually design protein-protein interactions, I think for many it's clear because nature has used them for a lot of tasks. They are one of the fundamental building blocks of life. However, they are also incredibly important for therapeutic development. If we just think, for example, about the PD-1, PD-L1 axis, which is very famous as a checkpoint inhibition, where you would have like cells of the body expressing PD-L1 to essentially tell the T cell through PD-1 that they should not be attacked. And then tumor cells essentially harnessing this mechanism, overexpressing PD-L1 to like hide themselves from the immune system. And people have developed agents, mostly antibodies, that would bind to PD-L1 or PD-1 and kind of disrupt this axis and therefore kind of making these tumor cells visible again for the body's immune system. And actually one of the most successful drugs in the world is Keytruda as an anti-PD-1 antibody. It's, I think, in 2024 was the most successful drug out there. So there's really like a motivation, also a lot of money involved in developing molecules that can kind of influence protein-protein interactions. And so if we can do this fast and efficiently from scratch, this would be great. If we want to design a protein, we need to kind of all get on the same page about the general basics of protein design and protein biochemistry. So in general, I will go through this quite fast, just to kind of update everyone. We kind of go from this amino acid sequence to secondary structure elements such as helices or sheets. And then we assemble this into like a tertiary structure, which then ultimately alone or together with other proteins performs a certain function. And this is kind of like nailed down in this Unfinsen dogma that sequence ultimately determines structure, which ultimately determines function. Now, it's very important, and one of the big questions is essentially how do we go from sequence to structure? Like, how is this kind of encoded? And for many, many years, we have been approaching this from like an energy perspective, and this is kind of where physics came in, where you would try to essentially calculate the forces holding together a protein and then kind of finding relationships between a sequence and a low energy conformation, and therefore a very probable structural ensemble. However, in recent years, of course, this has shifted a little bit with the abundance or like with the development of AlphaFold. We have made a huge step forward, and actually going from sequence to structure, especially for simple proteins, is actually something that is not so hard anymore. Now, in protein design, we kind of also want to go from sequence to structure, but usually it starts the other way around. You kind of start with a function in your mind. You want to bind X protein. You want to maybe interact with a certain small molecule. You want to block an interaction, etc. And then you know, okay, for this I need some certain structural constraints, and then you need essentially go back and try to find a sequence that could potentially kind of accommodate this function by folding in the structure you envisioned. And you kind of go back and forth between these two things to ultimately get to a solution that is plausible. For us as humans, it's actually very interesting to explore the space beyond what nature has done. So if you think about how big a protein, what the space of a potential protein of 200 residues is, you will very quickly come to the realization that nature actually has found a solution and then explored this essentially space around it. But it did not move very far because there was no evolutionary pressure to essentially explore the whole space if there was already a solution that worked well. For us as humans, because we don't naturally or we don't necessarily want to have only natural functions, there might be a lot of potential proteins out there in this space that could perform a function that we are interested in, be it, for example, enzymes, but also be it for therapeutic development. If we go through a standard protein design workflow, we kind of have these three branches that in the last couple of years have started to kind of become the gold standard, which starts all with kind of generating some sort of backbone that not necessarily has yet side chains and sequence information. It's more like to get a rough idea of the shape of a protein. If for a binder, it's usually kind of about how the shape complementarity for a target, for example, is. Or for other proteins, it's about the shape, the size. For example, for a membrane region, you need a certain length, things like this. And you can use tools like Rosetta, but mostly you would use things like diffusion models nowadays, like RF diffusion or Chroma. Then you need to find a sequence that folds this backbone that you just kind of roughly sketched out. Ideally already having kind of also the function in mind that you want to tackle. And for this, you can also use Rosetta, but more prominently protein MPNN and also kind of other things like Carbonara, but protein MPN really kind of is the big guy on the block here, and everybody in the field kind of uses it. And after you have kind of an idea about the structure, you now have a sequence that potentially folds into this structure. You might want to make sure that this is actually the case. So you do some sort of post-hoc filtering by repredicting the structure, the sequence that you just designed, and see if it matches the backbone that you had early on kind of sketched down. And for this, you can use things like AlphaFold, ESMFold, Rosetta Fold. There are many structure prediction tools out there that do roughly the same. Now, in the field, we call something like this the designability test, where we stick with RF diffusion for now, which is we make a quick protein, and then we assign the sequence, we repredict it, and then we align the two sequences on top of each other, and we see how close does essentially what we repredicted match what we designed in the beginning. And we can quantify that with TM scores, or many people will be familiar with just classic RMSD calculations. Now, if we go into now we know kind of how proteins are designed in general. So now we want to focus a little bit more on what we today want to look in, which is binding something. For this, we go from right to the left, not from left to the right. We want to bind something, so we need to look at kind of what is important is the tertiary structure of our protein and how it kind of shapes around the target site. And when I actually started to look into this problem, it was 2024, and there were two methods, let's say, on the market that were in this transition period, actually, from physics-based to deep learning-based approaches. So one of them was Massive. Massive was developed by Bruno, so that was kind of the reason why I came here, where the idea was, hey, we can break down the binder-target interaction, break it down to a surface problem, where we would essentially train a model to describe surfaces as a vector description with the geometric and chemical features like curvature, hydrogen bondings, polarity, etc. And then we would essentially have a huge library of mathematical fingerprints of small protein fragments, and we could just very quickly see if they match together. This you can think about as like you have a hydrogen donor on your target, then at the same position of the binder would have to have an acceptor. If you have a hydrophobic residue, you need a hydrophobic residue to kind of match the energetic favorability that drives binding. And once you identified these kind of small stretches from the synthetic library, you then use Rosetta. So this was kind of still using physics-based design to graft it into a synthetic library and reassemble a whole protein. This would usually give you something around 2 to 5 hits, I would say, out of a typical yeast library, which would comprise in our lab roughly 2,000 designs. So you still have to kind of screen a library of designs to get a hit. And then later, not that much later, but there was RF diffusion, which combined it essentially now, which moved completely away from these like libraries by generating the backbones that I previously described, essentially for every new target on the spot. So you would kind of really generate a backbone for a binder first, for the target site that you're interested in, which makes obviously things like shape complementarity, etc., much more easy to achieve. And then you would essentially design a sequence on top of it and filter it. And actually this filtering was what drove the development of BindCraft. So the Baker Lab published a paper about where they looked into essentially their old yeast libraries and the experimental results that they had, and kind of showed that when they used Rosetta to score the designs, the experimental success rate was relatively low. But if they would have filtered essentially the design pool with AlphaFold for like essentially asking the model to see if it thinks that the binder binds or not to the target, they could have enriched a lot their design pools for successful designs. And this was kind of you need to understand that during that time, we were speaking about 100 to 10 million of in silico designed binders, from which only a fraction was successful. So every kind of improvement was a huge boost in terms of help for the PhD students who had to screen all these things. So what we thought is, hey, if AlphaFold is really so good in discriminating a binder versus a non-binder, maybe we can already use it for design. So what we did is we looked a little bit into literature, and we actually found already some like work that people had been doing. Sergey Ovchinnikov is one of the drivers of this, where people looked into using these structure prediction models for design. And I will give you a brief introduction here. This should not be comprehensive. It's just to motivate you in case you're interested to look a little bit further into it. So the easiest is to use a structure prediction network for design is to just start with a random sequence, then do a prediction. Usually it's nonsense, but then to essentially see, okay, if we now do a mutation, does it get better or worse? If it gets better, we accept the mutation. If it gets worse, we do not, we try again. And we do this iteratively for many, many steps. And this ultimately can fold a protein. So we can ultimately go from a random sequence to a folded protein if we optimize for confidence, as represented here as like the confidence of the contacts in the contact map. But you also see that we needed 40,000 steps to get to there. So it takes a long, long time. It's computationally quite heavy to get to this point. So maybe there is something that we can do better. And actually what we can do is we can just change the representation and do essentially gradient-based backpropagation. For this, we formulate what we want to optimize for as like an error. So you can think about this as like you have a certain fold in mind, for example, 2 helices. Then you do your prediction, you see, oh, it's 2 beta strands. Then you overlay the contact map, and then you from there can essentially calculate a discrepancy, like an error. And this you can formulate as a loss function because you want to minimize the error, and you can use it to guide the sequence into a certain direction. And this you can do in 2 ways. You can do this first by always forcing the sequence into like a representation that is physically realistic, meaning 1 amino acid at each position. This is what we know, right? This is kind of how a protein looks like. Or we can do it in a resort, what we call relaxed sequence space, where we use probabilities at each position. So instead of saying, here is an alanine, here is a threonine, we say like, it's not exactly this, but something along the lines of, we think 20% it could be a threonine, maybe 10% an alanine, maybe, you kind of work with these probabilities instead. And by this, you have a much smoother optimization process. And then just at some point, you force it into the physically realistic one amino acid at each position. You can see this in the following two trajectories, where on the left, you have essentially one amino acid per position in the binder. And on the right, you have essentially these kind of probabilities that you play with and that you optimize into a certain direction. And you can see that we converge on the right side, and on the left, we just essentially stay into like in the noise and don't really move forward in our convergence. Now with BindCraft, because we actually want a proper binder in the end, and it needs to have a sequence in the end, we need to traverse from this kind of relaxed sequence space into this one-hot encoding, what we call, which is the physically realistic form. And we do this gradually by applying like a softmax function, where we essentially over time essentially take out the amino acids that make no sense at this position. So we kind of gradually move towards this one-hot encoding. And you can see this on the right here in the trajectory plot, where we first, where we see that we are first in this relaxed sequence space, and then over time we transition into this like one-hot encoding, which is what you see now. And we optimize for different things. We can optimize for model confidence, such as PLDDT, PTM, PAE, which are different metrics from AlphaFold. And we can optimize for shapes. We can optimize, for example, a lot of contacts, which would make a binder very compact. We can use a radius of gyration loss to make it pretty globular. Or we, for example, want more like a better sheeted binder, so we can play around with the secondary structure elements and use the negative helicity loss. And you can customize this a lot, and we will go into this later on, how this works. Now, we formulated this not only as like this hallucination step, and actually this hallucination step was already heavily researched by Sergey and implemented in Colab design, but we kind of optimized it into an end-to-end pipeline, because both Martin and I are actually wet lab scientists by training, and we kind of wanted to have this as a tool for fast binder generation. So it was more like for us primarily to have it as an end-to-end framework where we could press play the evening before, do experiments in the wet lab during the day, and then come back in the evening and look at some cool designs. So we not only used this like hallucination step that I just presented, but we also did all the downstream optimization and filtering and included that in the pipeline, where you would essentially, after this hallucination step, optimize the sequence with MPNN to make it better expressible in the wet lab and more stable. This is just something that we know out of experience, that these sequences work well. And then the final filtering using a different set of AlphaFold weights to essentially cross-validate our sequence and compare it to the initial trajectory, doing essentially this designability test that I introduced in the beginning. And this whole pipeline is what we call BindCraft. Now, if you look at the trajectories of these binders and how they look like, you can see that these binders that come out of the pipeline can actually look very different. You have here on the left the purely helical protein, which, to be honest, is how most of the mini proteins look like. And on the right, you have like a purely beta-sheeted binder, which is more like a rare case, but you can enforce it. And we brought these designs into the lab, and actually they had quite a high experimental success rate, which was also much more than we expected. And we did this on over 12 targets to essentially see if it's generalizable. And in nearly all of the cases, we were able to, like from ordering 10 of these designs that passed the filters, to fish out one at least that had nanomolar affinity, which is usually a good starting point for many wet lab applications. And we then continued on many of these designs with amazing collaborators that took them and put them into a biological context. Now in 2026, there are obviously many other pipelines around, and the field is moving incredibly fast. But I just want to say here now, the modern tools that are coming out, they are mostly about either refining a little bit the success rates, even though sometimes it's more just like an like a parallel method that is like an option for the user, but also to expand on like the involvement of small molecules, of conformational control, and especially kind of getting antibody-derived formats into the designable space, which with BindCraft is so far pretty tricky. So there are many out there, many of these tools. However, the lessons that you learn for one of these pipelines, that's why I'm mentioning, is very transferable to all the others. So it's good to spend some time on one of these pipelines, and oftentimes you can then kind of understand a lot easier how the others are working. As I said, it gets more and more important to actually think outside of a binder is just a pure binder. Many people think about integrating them into complex things like CAR T cells or viruses, or even using them as scaffold-defining elements in nanomaterials. And oftentimes we even add a layer of control to them, like light responsiveness. You have a drug that induces the binding, etc. So we kind of make good progress in making this very modular, very efficient, and adding additional layers of external control. Now back to the kind of the basic, the boring stuff. I want to walk you through one of the applications to motivate you that you can now, with these tools such as BindCraft, really go from binding to function to like an initial kind of hypothesis proof in a few weeks or months. So we got approached by a long collaborator of our lab, Alex Vecchio, from the University of Buffalo, who kind of works a lot with Claudines. And Claudines are very involved in one of the mechanisms of a toxin from food poisoning, which is released by Clostridium perfringens. It's this blue toxin that you see here on the left that bind to Claudines. They form huge pore, like oligomerize them, and then which ultimately leads to a cytotoxic effect. And he asked us, hey, with your pipeline, could you now make binders that block this interaction so we could rescue the cells? And that's what we did. And we could actually build on work that Morten has been pushing before, which is bringing membrane proteins into the soluble space. And there they also, just by chance, also worked on Claudines. They are quite simple molecules because they have only a few helices in the transmembrane region. So what they did is to, and I don't want to take too much away from their work, they essentially fixed the epitope, which is this essentially palm region on the top, and solubilized the rest. So you could now use it as like a soluble protein. And we had two flavors of this, one that was a bit more conservative than the other. So we could essentially just express our binders in the wet lab and first check for binding against these soluble versions, because we had to deal with the very hard to work with membrane proteins where you have to detect detergents, etc. So we quickly checked a couple of our binders against these soluble analogs, and we saw that some of them actually, here's binder 12 as an example, bound against the soluble analogs. And then we sent them over to Alex, and he checked them against the wild-type Claudine, now with like a restricted set of binders. And he could show that they would first bind the same binding site as the toxin, but second, could also kind of compete for the binding site. And this was very important for the next step, which was to essentially now do kind of this survival assay where you would treat cells with the toxin, which would usually kills 75% of your cells in the dish, and then essentially look in comparison to a known toxin inhibitor, how do our binders perform in rescuing the cells? And you can see here that essentially with different concentrations of our binder, we can rescue nearly as many cells as the literature known and optimized toxin inhibitor. So, and this took a couple of weeks to months. A lot of this was shipping and kind of coordinating the project. So what I'm trying to tell here is you can use these methods to really probe a biological idea very fast. And I think this is the beauty here. If we can really compete with antibodies and on the long run, I'm actually not so sure, but they can be a really good research tool to do these kind of like experiments. Now to switch a little bit track and to also move towards a little kind of tour through BindCraft, I want to introduce you to my actual main PhD project, which is to essentially modify viruses for advanced therapies using de novo design. And it's mostly about adeno-associated viruses, which are the most prominent gene therapy vector out there. They are super cool, but also incredibly expensive because they are quite hard to manufacture. You need a lot of them because they go everywhere in your body and you only hit with a small fraction on target. They are quite immunogenic, so you have like a one-shot therapy because it's a protein capsid, so your body will develop antibodies against the virus. And because of all these things, we have only or mostly therapies for rare diseases out there where we do gene replacement therapies, where off-target effects are not bad because you bring in a healthy copy of an already existing protein. But because of the small sample groups that you have in rare diseases, the treatments are incredibly expensive and also actually quite a burden for many healthcare systems. So what we thought about is if we can increase the specificity of these viruses, we first might need less virus to inject into a human. Second, it could be a great research tool. And third, we might open these vectors up for treatments that have a broader sample group, which will ultimately drive down the cost of these therapies. And this is in collaboration with Christian, who is another PhD student from our lab, and Bernard from the gene therapy platform in Geneva. And we essentially took one of these wild-type AAVs, which usually kind of go into any cell, and first of all knocked them out. So we removed all the interactions that they have with cells. So they are essentially silent viruses. And then we introduced binders that we had designed into the capsids and kind of rendered them now active again against a certain cell surface marker that we chose to be only present on a certain tissue type, such as cancer cells or lymphocytes, for example. And these binders, they need to have certain properties. First of all, they need to be small. Second of all, they need to bind to the target, but also they need to have the N and the C terminus close together to be essentially serve like a plug and play platform that we could repeatedly use without having to optimize every single time, like linkers, etc. So if BindCraft could make these binders already with these properties, that would be great. And this brings me to kind of the part of the workshop that is a little bit more hands-on, where I will walk you through how we could set up such a run to make these binders for AAV redirection. And we first start with kind of the requirement that we need a binder for binding to PD-L1. So what you do is and this might now sound very trivial to many of you, but I think it's always a good refresher for the people who have not worked with protein design a lot. You go into like the PDB, you search for a crystal or cryo-EM structure of your molecule, of your target of interest, or if it's not present, you can also, of course, use the AlphaFold structure. And what is very important for any protein design tool is that you crop it to the part, usually the domain that you are actually interested in, because your runs will, in terms of compute time, will scale with the size of the system. So the smaller you can break it down to the problem that you're actually interested in, the better it becomes, and the more efficient it becomes. And for PD-L1, we were interested in essentially looking at the binding site of PD-1, which you can see here on the bottom right, in purple and rose. Now, for PD-L1, it's very easy because you can just crop out this domain. For other proteins, it's actually quite tricky. But you can actually provide BindCraft, and this is thanks to some really cool helper functions that Sergey implemented into Colab design, come with very unrealistic croppings of a structure. For example, you can see here on the right, this is sliced out of a trimer, and we were interested at the interface on the top. So we didn't need a lot more context. So we just sliced it down to these two essentially subunits. And for example, for membrane proteins, you can remove the full transmembrane region, etc., which all speeds up the process. And this is actually cool. The only thing that you need to do is you need to, when you run BindCraft, look at, is my run stable, and is the confidence of the target in AlphaFold still sufficient for design? Sometimes if you crop it too much, AlphaFold just says like, okay, I have no idea what's going on anymore. And you would see that in the confidences of the target. Now we have our cropped structure. We could use either the two IG domains of the PDL1 or only one that we are interested in. But we also need to define on these domains the kind of residues that we want to interact with. And there are actually two different things that you can do. First, you can let BindCraft choose. Then what it will do is it will just sample all over the place and will optimize for as many contacts as possible. Or you can specify hotspots, and then you will BindCraft will optimize for contacts with these residues that you kind of provided. And in comparison to other tools where one hotspot is usually enough, here it's really like you want to give it 5 or more hotspots, because then this like contact optimization is much more efficient than if you only provide one. It can be enough, but it's better to have 5 or more. And then you kind of can write down these hotspots. In this case, what I did is I just looked at the PD1, PDL1 interaction and took the ones out from the interface. If you have no clue about kind of, you have a new protein, it's not well studied, what to do, there are other tools that can help you. There is an example from just a group close by from the Del Ferraro lab that developed Pesto, which is essentially a tool to provide you with, given a protein structure, what residues on this protein structure are most likely involved or well suited for a protein-protein interaction, or a protein ligand or protein ion, etc. But for us, the protein-protein is the important part. It's a web server, it's super easy to use. I don't want to make advertisement here, but I use it a lot, and I just highly recommend it. And you can see, for example, if we now look at the Pesto prediction for PDL1 and what BindCraft is sampling, if you don't give it hotspot, sometimes it just matches and AlphaFold kind of gets it. But for some other times, if you don't provide a hotspot, it just samples all over the place, which either gives you kind of binding sites that have never been explored, or it is also unrealistic. So it is kind of a bit this trade-off of like guiding versus non-guiding. Now you're actually set, because you now have a PDB structure with your crop thing. You have your hotspots written down. So the only thing left to do is here to kind of prepare all the files that you need for a BindCraft run. There are actually three files that you need to run BindCraft after you have successfully installed it on your cluster or wherever you are running BindCraft. And there's this target setting file. And this is really kind of the basic file where you provide all the paths. You can define the hotspots. You can define the length of your proteins that you want, like the binders. And you can define the number of final designs. That is essentially how many wet lab ready designs do I want essentially out of this whole pipeline. And then it will run until you reach this number. Then there is the second file, which is called the advanced settings file, which is one that I encourage to look into, but I disencourage you to change anything in the beginning when you kind of start to play around with BindCraft, because here there is a lot of like fine-tuning, fine-tuned weights and sample steps, etc., that we have predefined and optimized. And you can change them, and sometimes it helps, but most of the time the default settings are working well. So it's good to understand what different things here are doing if you go through the code, but I would disencourage you from changing anything here. And you have different sets of these advanced settings for different problems, such as peptides, hard targets, flexible targets. If you only want beta-sheeted binders that you can find on the GitHub and that you can just use out of the box. Then you have a filter setting. So this is essentially for the last step of BindCraft, where you repredict all your designs, what you want to filter them on, because you only ideally want to take the things into the wet lab that have a high probability to work. So you really kind of want to filter them properly using these metrics. Again, I think the default filters are good. Sometimes you need to relax them a little bit, or if you have fantastic results, you can also increase them, of course, to only get to really high scoring designs. And then the only thing that is left to do is to essentially write a small submission script, and then you can just submit it on your cluster using these three files that we have just prepared: the target settings, the advanced settings, and the filter settings. I have here added a notebook QR code that will bring you to the BindCraft Colab notebook, which is incredibly slow because somehow the implementation on Colab is not as great. However, it's a fantastic starting point to kind of play around with the settings and understand the basic concepts. You just need to be a little patient. Now, if you have run your designs overnight and you come back the next morning, what you will hopefully find is these kind of outputs. You will find folders that will give you the trajectories, MPNN, and then accepted and rejected designs. So the most important part is the accepted designs. In there, you find designs that have passed all the filters that you have been applying, that in theory are ready to be ordered for wet lab validation. In MPNN, you find essentially one step before, essentially just the repredicted MPNN sequences, not yet filtered. And then in trajectory, you can find the raw trajectories before sequence optimization. In these folders, you generally only look if you don't get anything in your accepted folder and you want to do troubleshooting and you want to see kind of why does it fail. And you get guided by this by a CSV file called failure CSV, which essentially tells you because of which of the metrics and at what stage did most of your designs fail. So this is a good overview to kind of start troubleshooting. And then again, final design stats is the CSV that is the most important. That's where all the good sequences are stored in. Now, if we go and show some of these PDBs, they actually look pretty decent. This is now for PDL1. We get mostly helical binders, and they nicely engage with our target surface. However, one of the problems that we see is that the termini for many of these designs are still kind of on opposite sides. So we would have to introduce huge linkers into our AAV if we would want to plug them in. So what we can do is we can now adjust BindCraft, and this is now really something for the people that have spent some time on the pipeline, and you can write your own loss function that essentially now optimizes for the distance between the N and the C terminus of the binder. And we can essentially now incorporate this functionality or geometric constraint already in our design process. And this really kind of can speed up finding binders for certain applications, but also, of course, requires a certain amount of dry lab skill or some help or just some try and error, which usually nowadays is actually quite achievable. It's not super tricky. And you can implement then these losses into BindCraft, just add them to the general loss functions that we have already implemented. And then you need to give it a quick benchmark where you essentially look, how much do I give a weight to this loss function in the optimization process? You can see here on the x-axis different weights that we have added for this NC terminus loss. And you can see that the more weight we give, the shorter the distance on average becomes between the two termini. So we are really optimizing for the right thing. And if we now look at some of the results with this loss function, we can actually see that now our termini are close together. For example, in this design, they are very nicely together, so we can just plug and play them into our AAV. We can also, as I said before, for example, change the secondary structure. Here we have, this is also already implemented in BindCraft, helicity loss, where we penalize the formation of alpha helices to get beta sheeted binders. And you can again see that we, with a loss weight of, for example, -2, we get nearly purely beta sheeted binders. Now we can plug them into our AAV, and to kind of round up this walkthrough through the project, I just want to give you a small outlook what we have been doing in the wet lab with it. we expressed now these AAVs with the binders in there, and we did this and tried them on different cell lines that expressed the receptor of choice that we want to target and an off-target receptor. And the AAVs contain a GFP genome, so if they go into a cell, they would over time express GFP. And in this plot, you can see that we have been doing this for quite some designs, some directed against PD-L1, some redirected against HER2. And you can see that in comparison to the wild type, some of our designs have now a specificity for the respective target, which is what we want to achieve. All of these binders come straight out of the computer. They have not been pre-screened for binding. They really go from the computer into the virus. And we have since then optimized one of the PD-L1 viruses, which now on hex cells, so on a very artificial overexpression system, outperforms the wild type AAV. And we can compete the transduction of the virus with an anti-PD-L1 antibody to really show that we are dependent on this interaction that we designed. We can put them onto an SBR chip, do some biophysical characterization, put them on mass spec. So really we can do a lot of cool biophysical assays with it. And the most convincing is, I guess, these pictures here where you can see that our PCS 395, which is our best variant so far, is really selective for PD-L1 overexpressing cells in comparison to the wild type virus that goes into any of these cell lines. Now you might ask yourself, okay, I don't have a cluster, what do I do? Generally speaking, this will hold you back, but there are some things that you can do, and this is something that has been pushed by a collaborator of us, Aaron. I don't want to do any advertisement here for any of his or other people's platforms. I just want to present this to you as an alternative, because the command line working with Linux can be pretty tricky. So there are these web-based tools like Ariax, like Tamarind, like Levitate, where BindCraft has been pre-installed, given that the community has requested it, and where you can essentially pay for both the service of the website, but also for the GPUs. So this is good if you don't have the infrastructure from your university and you still kind of want to dig into it, and you have played around with the Collab Design Notebook and now really want to launch your first design campaigns. Now, there are some considerations and observations for BindCraft that we have made postdoc. One of them being that the success rates are dramatically different in silico for different targets. For example, PD-L1 has been a fantastic target. It was very easy to get passing binders. Passing means going all the way through the pipeline into the accepted folder. Versus others where we had to sample a lot to get to this point that we had enough to order for wet lab validation. So sometimes it's really all about having 2 or 3 targets in mind, trying them all, and then maybe for the beginning focusing on the one that is the most efficient in silico, if you have the choice. Also, the binder length, the most ideal binder length can vary between targets. So sometimes small binders are better, sometimes big target binders are better. This is very tricky to predict. So sometimes it's good to maybe batch it into different length batches and kind of see which is the most efficient one, and then sample in this space to be a little bit more organized. And what we also see is that we have this kind of induced fit behavior, because in comparison to the fusion models, where you have like a rigid target, we co-fold also our target every iteration. So the target can also adapt to the binder as much as the binder can adapt to the target. You can see this this is also different from target to target, what you can see here in the RMSD plot on the right. We can also force this. There's also like the so-called flexible settings, where we kind of mask certain parts of the input structure, and therefore AlphaFold is forced to fold more things by itself rather than relying on the template information. And with this, you can essentially increase this flexibility during design. Now, one of the important considerations when you choose a target site and want to evaluate if something is proven to fail or if it has a high chance of success is really to kind of look at your protein and do a pre-assessment. In general, hydrophobic interactions are driving binding. You don't want too much of them because then it becomes unspecific. You don't want too little of them because then you don't get enough binding energy. So it's a little bit of like a good mix. But yeah, if you have a fully hydrophilic site, this is usually much harder to target than a hydrophobic site. Flexible residues, if you have a lot of them in your binding site, it will also be more tricky. So lysines, for example, are not so great. It's easier to bind to structured regions than to loops. And usually it's more easy to bind to monomers rather than multimers, even though with the right setup, you can really optimize a lot there with the cropping and the right input structure to get also multi-domain input structures to work, multi-chain input structures. Of course, BindCraft has its limitations, and I don't want to scare away from this. One of the big frustrations that you will run into is that it takes quite some time. I want to say that this can be incredibly annoying because the feedback cycle is not as fast as, for example, for diffusion models, where after a few seconds you get a first design. But on the other hand, we have benchmarked this excessively. Oftentimes, what you lose in speed, you gain in quality. So in the end, if you say like, I want 100 good designs, you spend more time per designs, but also essentially it's well invested. While with many of the diffusion models, even though they are also getting better and better, you usually generate thousands and thousands of in silico structure and then need to filter them down to just a couple that actually have the quality to be validated in the web lab. And then something else is that sometimes AlphaFold does not respect this like hotspot optimization, especially if you have large hydrophobic regions. For example, if you use a membrane protein, if you put in a membrane structure, oftentimes find that it wants to bind to the transmembrane region, which makes sense kind of. But then it's hard to get binders to the actual epitope. The best solution here is to just cut away, essentially chop the membrane protein like a tree and really only design against the extracellular epitope and remove as many of these hydrophobic parts as possible. Now, some last remarks on the wet lab validation. I want to go through this quite quickly before I'm happy to take any questions from you. So this is now very basic, but I just want to give you an idea on what we have been doing. So we have been doing most of our experiments with like standard E. coli expression and purification. We usually did 250 milliliters of culture, so not a lot. So you can really get a lot. These proteins produce incredibly well, usually a couple of milligrams per liter, if not a lot of milligrams per liter. We usually do both affinity and size exclusion to get very pure results. However, of course, you can adjust this based on the requirements that you have for your assays. We use, for most of the experiments in the BindCraft paper, we have used SPR, where we looked at where we screened all the designs of the purification against the on and an off target, and then looked at like yes, no at 10 micromolar. Now, this is an arbitrary number that we have chosen based on what we kind of had in most of our tubes. But I think it's a good starting point. You can, of course, also use other things like BLI. There's also immobilization-free things like flow dispersion analysis, or you just look at it in a pull down or something. So there are many versions, but I really like kind of to get some binding curves from the BLI or SPR. And this can look like this. On the top, you see, for example, like an SPR plot, on the bottom a BLI curve. And one thing you can always do is to make sure that your binder binds at the right spot is to essentially have a competitor and then do like a competition assay with them to make sure that your binder binds at the right spot. I have to say that for so far, I have not yet had a binder that bound at a different spot, and I found out essentially. So I'm quite positive, but it's always good to check. You can obviously check for off-target binding by doing it against similar receptors, and you can probe like their oligomeric behavior. We see that quite some of our binders are dimeric, but they bind as monomers. So this is quite an interesting phenomenon. And we see that most of our binders are essentially stable up until you boil them. Like they are really stable molecules. They also crystallize quite well. So oftentimes you have good chances to get them crystallized, especially at low pH, given their high negative surface charge. With this, I want to close into like a future perspective slide, saying like, hey, what we actually need now is we can actually discriminate binders from no binders using AlphaFold quite well. But what we need is actually better metrics that are predictive of affinity, because for many applications, we need more than mid-nanomolar affinities. We also need to have like community standards on what defines a hit. For example, in RF diffusion, they looked at a positive control and said like, okay, a hit is if it reaches 50% of the response. We define it as a binding signal at 10 micromolar concentration of the binder, as we didn't have controls for some of the targets because they were completely new. There is something out there. Again, I don't want to do advertisement for any companies, but Adaptive, a company just here in Lausanne, they have, together with us and together with others, come up with a set of protein targets that is easy to work with, but also kind of generalizing a set of targets that you could, if you develop a new binder design method, probe your method on and benchmark it. And with this, I want to say thank you to all the amazing people involved in this work. Like we have a huge lab, and many people have helped us to characterize all these things, all the collaborators, all the facilities, and of course, thanks to you for giving me the chance to talk here. And I'm happy to take any questions.
Thank you so much, Lennart. Super interesting presentation. Extend to you a virtual round of applause on behalf of all the audience. Now we are indeed ready to take questions. You can raise your hand and unmute yourself to ask any questions you want, or you can write them in the chat and I will read them out loud for you if you don't want to unmute yourself. In the meantime, while the audience comes up with questions, I can start maybe. First of all, it's really impressive that there is a tool that allows almost anybody, of course, who needs some computing power, but still to design de novo proteins. And about the novelty of the proteins, I wanted to ask you how indeed can BindCraft go far from the protein structures where it was trained, and at which steps do you think there is the bigger bottleneck in how far you can go, like in the first generation, in the evaluation? What do you think?
Yeah, so the novelty actually is, even though it doesn't look like it, because many of these binders just look like helical bundles, but if you quantify them, so we did quite a lot of benchmarks on it to answer exactly this question, where you look at the TM scores against like a set of proteins from nature or from synthetic libraries, and actually they are pretty novel, so you will not find the same protein twice most of the time. However, they all have similar like features, which is mostly this high secondary structure content. And I would also say that this is kind of the bottleneck. So most of our binders are not interacting with loopy regions, which is something that antibodies, for example, like to do, which oftentimes has a different kinetic profile in terms of binding than what we have when you interact with these super pre-organized secondary structure elements. So I think this is a bit of a bottleneck, and we have tried to push it into this direction, but it's still a challenge.
Okay, thank you. I will read some questions that came up in the chat. First one by Elena. What's the next step towards turning any of these binders into real products or drugs? Is it even really doable to enter a clinical pipeline?
So I think it's absolutely possible, and actually it happens already. There is the big question of immunogenicity of these molecules, and also why you will find many of the pharma companies being a little bit scared about these new modalities, because for antibodies, we have over the last couple of like decades developed strategies to essentially deal with many of the challenges that come during a drug development campaign. But given that every protein that comes out of these pipelines is new, you essentially don't really know where to start. But there are first tries. And funny enough, some of these trials look very good. Oftentimes half-life is a problem rather than immunogenicity. But there is a very limited data set so far. So it's hard to tell what is really missing. I think it's just people having the courage to try. And so we can see where the bottlenecks really are. But yeah, for many things, you don't need picomolar interactions. Sometimes you do. So you could use some of these molecules and think about right formulation and try it, usually in an animal.
Thank you. Second one is by Tammy. How can BindCraft be leveraged to design proteins that not only bind a target more effectively, but also reprogram biological interactions in ways that reveal new functional capabilities beyond natural protein evolution?
Yeah, that's a very good question. So I think it all boils down to how much do you understand your system. I think if you understand your system very well, for example, you know that a certain cascade of interactions is pushing a receptor into signaling, you can, of course, either push for this function or you can avoid it. So like a bit like the agonism versus antagonism conversation. The problem is always that in order to design something that does something new, you need to know you need to design to achieve it. I think if you know it well, you can formulate it in some sort of geometric constraint or loss function to get closer to this goal, but you really kind of need to know this geometric constraint in order to be able to guide the design. So it's a little bit this trade-off.
Thanks. By BT Kelly, first of all, great talk. When cropping a target membrane protein structure, how would you deal with non-consecutive parts of the sequence that fall together to form an extracellular domain? Connect them with soluble linker?
No, you can really just remove these residues, for example, in Pymol, and BindCraft will deal with the essentially chain breaks. It is okay if there are multiple stretches that do not form a complete protein, but you can really kind of go to extreme crops to minimize it to the system you're interested in. So don't be scared about these chain breaks.
Okay, impressive. A question by Hamed. Can we get picomolar affinity with these binders like what we can get with antibodies?
So first of all, I think we need to be a little careful to always compare an antibody. So definitely many antibody-derived formats, if they have been screened from libraries, come with higher affinities. But if we look at pure antibodies, they also come with some sort of avidity. So we should not forget about this. However, you are right that even the FAPs or the SCFVs or nanobodies come with high affinities if screened well. I think it's possible, and people have shown that you can, for example, take binders like this and do some sort of evolution campaign on them or site-directed mutagenesis. So this usually still involves screening, but the field is aware of the problem, and I think people are trying to come up with strategies to do this in silico affinity maturation. You can reach these goals with excessive engineering. However, it's unlikely that you get a picomolar hit from your initial design. That's so far, I think I have not gotten any without avidity being added to the design. Yeah.
All right. Thank you. Again, super interesting talk. I think we are right on time. So I would like to thank you for joining us again and for participating to the discussion. Thanks to the audience as well for the questions.